start, we would like to acknowledge that today we are dialing in from the lands of the Wurundjeri people who have been custodians of this land for thousands of years. We would like to acknowledge and pay our respects to their elders, past, present, and emerging, and extend this respect to everyone who may be dialing in from different countries. So I invite you all to use the chat function to write a quick acknowledgement of country of your own if you'd like.
As you probably already know, UCCC is an initiative of the Young Australian Cancer Initiative. We are a student-led non-for-profit organisation that aims to nurture a future generation of cancer-literate individuals who are empowered with the skills and knowledge to reduce the cancer burden in our society. The UCCC aims to provide an opportunity for university students across Australia to get together in groups of 2 to 4 to tackle a real issue in the cancer space. This year, the case challenge is to design an intervention to increase HPV vaccine 2-dose course completion amongst First Nation adolescents.
It is now my pleasure to introduce our workshop facilitators, Hasna and Hashini. Hasna and Hashini are esteemed tutors from YACI's High School Masterclass program. I'm sure we're all very excited for them to show us how cancer unfolds. So without further ado, I'll let you take it away.
Okay, great. Give me 1 second. Can you guys see my screen?
Yep.
Okay, perfect. Okay, so I'd just like to introduce myself. As Aisha said, my name is Hashini, and I'll just be taking you through a couple of the concepts we'll be covering in today. So I'd just like to start off by saying a couple things just to, I guess, get ourselves sorted. So if you guys have any questions, just chuck them in the chat, and then Hasna or I will try to get to you as soon as possible. And if not, we can always organise after today's workshop. And then today we will be covering quite a lot of content. So I think the best way to go around is probably just to listen and just listen and relax today instead of frantically taking notes, because I think you'll absorb it more if you just get your head around the concepts, and then later on we'll give you the slides and you'll be able to just understand the concepts in a bit more detail. So I'll just go through an overview of what we'll be covering today. So we'll be covering an introduction to cancer, the cells and the cell cycle, basic cancer biology, the hallmarks of cancer, HPV, cervical cancer basics, prevention, and then the treatment options of cancer. So let's get started then. So basically, what is cancer? So all organisms are basically composed of cells, right? So some of you may already have a general idea of what cancer is. The topic we'll be covering in your competition is cervical cancer, but we have many other types of cancer. We have prostate cancer, breast cancer. But the thing about all these cancers is that they have a general, I suppose, in terms of what they look like, where they originate from, we kind of can give it a similar definition. So if we were to give a definition to cancer, we can say that cancer is a group of diseases that are caused by abnormal cells that grow uncontrollably due to genetic abnormalities. But first, to understand the more complex aspects of cancer, we probably need to understand what kind of makes up cancer cells and how they're able to grow so abnormally. Okay, perfect. So first of all, we'll go through an introduction to cells and the cell cycle. So all organisms are composed of building blocks called cells, and these are considered the smallest units of life. So think how bricks kind of make a house, Lego bricks make a Lego house, and now think of something a million times smaller and things that make up humans themselves. So humans are composed of many different types of tissue, which in turn have many different types of cells. So if we go with epithelial tissues, so this kind of lines the surface of the body and the organs. So think of your skin, it's lining all our, kind of lining the external surface. And then we also have connective tissue, which kind of connects the tissues within our body. So the most common one is adipose tissue, which is also known as fat tissue, but we have other ones, such as cartilage and bones even. And then we can move on to muscular tissue. So that's involved in contraction and relaxation. So any movement you're doing, any movement I'm doing right now is all due kind of to muscles and the muscular tissue. It helps me flex and extend. And we kind of have types of muscular tissues as well. So we have skeletal tissues, smooth tissues, and cardiac tissues. So cardiac would be to the heart, smooth tissues is usually due to involuntary movements, and then skeletal is more for voluntary movements. And then finally, we have nerve tissue. So this is to do with nerve impulses. So basically, when we see something, when you're reading your slides, you're kind of processing it as external stimuli, right? But then your nerve impulses is kind of what helps your brain, I suppose, understand what you're seeing. So, of course, different cells in our body kind of have to perform different functions. So, everything you've seen right now, they're all performing different functions. And this kind of allows us to do multiple things at once. It allows us to, I suppose, just coordinate movement, generate heat, and do a lot of things. But the thing about all these cells is that they have similarities with one another. So in terms of similarities, we can think of this as like the cell ingredients. So what we have in common as humans is the components we have in our actual cells. So if we go, I suppose, from the boss of the cell. So think of the nucleus, it's like the cell's brain. So like how our brain kind of controls our thoughts, the nucleus kind of tells all the other organelles. So we call the components organelles. It tells all other organelles what to do. It coordinates cell activity. And then moving on from the nucleus, think about how you probably have ate, actually, I don't know if you ate dinner by now, but you probably ate lunch today. So you ate lunch to get energy. So that's kind of what the mitochondria does. It acts as the cell's energy supply. And then with this energy supply, we can get to work. So some of you might have gone to university today, some of you might have part-time jobs. So we all work, and that's what we call the endoplasmic reticulum. We can consider it the cell's worker. So we have two types of endoplasmic reticulum. We have the rough endoplasmic reticulum, which is right here. So it has like stuff studded onto them. So we call these little dots ribosomes. And what the rough endoplasmic reticulum does is that it helps make proteins, which we use to coordinate cell activity as well. And then kind of the smooth one, so the one that without all these like protrusions coming out of them, is the smooth endoplasmic reticulum. And the smooth endoplasmic reticulum has its own set of functions, which the most common one is that it makes fat and adipose. And moving on from that, we kind of have the cell polishers, so the cell packages. This is kind of what makes our proteins really able to function. It helps packaging proteins for export in the cell, export out of the cell, or we can also use the proteins in the cell. And finally, you know, when you've done a lot of work, you kind of have those rough drafts. You kind of have a lot of like leftover rubbish. So we kind of put all of that into a bin, right? And that's what we call our lysosomes. So the lysosomes is kind of like the garbage dump of the cell. It's what we call our waste removal system. So we'll go on to the nucleus. So the nucleus is, like I said, kind of like the boss of the cell. It's like, I suppose, the most important part about the cell. So it contains our DNA. And I hope some of you know that our DNA is kind of what makes us all unique. It's basically our genetic material. So it also has a couple of other functions as well, though. So it's the first site of protein production. So we'll go into this a bit later, but it's where transcription occurs. And it's also where cell replication occurs, and obviously, as we said before, it coordinates cell activity. And yeah, just note that its DNA stands for deoxyribonucleic acid, and that kind of helps when we go into the more, I guess, niche part of DNA. So if we start from the smallest aspect of DNA, we have what we call the nucleotides. So we have kind of a ball, which we call a phosphate group. We have a pentagon, which we call the deoxyribose sugar. So that's where we get deoxyribose from the nucleic acid. And then we have what we call a nitrogenous base. So the nitrogenous bases, we have four types. We have guanine, cytosine, adenine, and thymine. So as you can probably see on this picture, we kind of have a pairing system. So guanine in red pairs with cytosine in blue, and then adenine in yellow pairs with thymine in purple. And this is kind of what's unique about the cell, is that we have this pairing system, and then we form hydrogen bonds in between. And then, yeah, moving a bit for more of a bigger picture, we have genes. So maybe some of you know that we have genes that we kind of inherit from our parents. And we can have different types of genes. So I have genes for, I think my eyes are like black or brown, but then some of you might have blue eyes. So the genes are what causes all these like unique differences. And yeah, moving on from going even bigger is the chromosomes. So in humans, we have 46 chromosomes in our body cells, and this is really what we inherit from our parents, and it's what makes us unique again. So why is this sequence kind of important, and how does it relate to cancer? So the DNA sequence is read in order to build proteins. So it's kind of like a specific sequence. So the central dogma of life is that the nucleotide sequences we were talking about before, adenine and thymine, guanine and cytosine, it encodes for like a sequence of proteins. It encodes for the structure of proteins. So obviously, if the sequence is wrong, the protein we make is kind of incorrect. And the proteins have specific functions. So if these proteins are incorrect, they can't carry out normal cell functions, and that's when things like cancer and other bad things occur. So we might go on to, we'll go into now how we kind of make these proteins. So we have it's kind of like a two-step process. So as we said before, transcription occurs in the nucleus. And basically, if you think of one piece of paper as your DNA, if we rip it into two, we're kind of unzipping it. So we can unzip the DNA or rip the paper in half, and we have a top and a bottom. So we do that with an enzyme. So RNA polymerase is what unzips the DNA. And then we kind of do a couple more things to it. So we add a mRNA to it, and then we kind of just make it so that it can be taken out of the nucleus and taken out of the nucleus and going out of the cell. So we have the cytosol, which is kind of like, if we go back to, okay, so think of let me go back. Okay, so this part is like the cytosol. So once we go out of the nucleus into the cytosol, we can try and find a ribosome, and that's where translation occurs. So translation is ultimately what kind of forms the protein that we'll be using to carry out cell functioning. So yeah, in summary of, I guess, the basic outline of cells is that we have different kinds of cells, and then the most important part about the cell is probably the nucleus. And then if we have the wrong proteins, we can impair normal cell function. So that's what's the most concerning part about mutations in the cell. And yeah, if you guys have any questions, just chuck them in the chat. And yeah, so we'll go on to cell cycle. Maybe I'll ask you guys a question. So where does cell replication occur? Does anyone want to unmute and just say? Did someone say something? Sorry, I couldn't hear.
In M?
Yeah, in M phase. Perfect. Yeah. So we have kind of two phases. We have S phase and then we have the M phase. So we'll start with the S phase. So interphase is where we're kind of just making the DNA bigger. So we're duplicating the DNA, we're getting the chromosomes to thicken, and this is so that later on we can split the DNA. So now we'll move on to M phase. So as I think someone said before, this is where DNA replication actually occurs. So we call it PMAT. So P for prophase, M for metaphase, anaphase, telophase, and then we have an extra bit that doesn't really fall under PMAT called cytokinesis. So with prophase, we have chromosomes chromosomes having two chromatids, and then we kind of dissolve the nuclear membrane around it. And then in metaphase, these chromosomes line up along this kind of, we call it an equatorial plate. So think of like an imaginary line, and then your chromosomes just lining up along the line. With anaphase, think A for angry, we kind of split those chromosomes into sister chromatids. And then telophase is when we're starting to like make the cells their own. And then cytokinesis is finally when we get these two daughter cells. So we have the parent cell dividing into two daughter cells with the exact same DNA. We've basically made a clone of the DNA. But the thing is, how does a cell know when to divide? So we have this kind of three-step process. We call it signal transduction. So your cell kind of gets signaled from something. It can have an extracellular signal or an intracellular signal telling them, oh, we want you to divide now. So we call that reception. It binds to a receptor and it sends a signal telling something to happen. So we say transduction can occur. Either we stimulate more proteins to do some extra activities, or we can straight go to the nucleus and say, can you replicate your DNA now? Can you make new proteins? And that leads to a cell response. So in this picture here, we can see that the cell response is more protein production. But as we said before, there is a problem with this. So errors can sometimes occur when DNA is replicated, because the wrong DNA can lead to the wrong RNA, which leads to the wrong protein, and finally abnormal cell production. We try and fix this. So we try to do things to stop this from happening. Your body is quite developed, so we try to either repair the DNA or we think to kill it before it can do anything worse. So with apoptosis, we call this programmed cell death. It's basically your body telling your cells that this cell is damaged, we can't fix it, it's irreparable, so let's just kill it. But sometimes with cancer cells, with some cells, they kind of evade both of these mechanisms and they keep on replicating themselves. So this leads to an uncontrolled cell cycle or uncontrolled transcription, and that ultimately causes cancer. So DNA repair. This repair is always by enzymes. So think about how RNA polymerase was unzipping the DNA. We have other enzymes which kind of repair the DNA. And as we were saying, apoptosis tries to fix this as well. We can just kill the cell before it gets any worse. And then we can also have our immune system try and kill the cell. So we have this thing called phagocytosis. So it's kind of like how your lysozymes work. We try to just kill the cell by just trying to engulf it, make it into little bits, and then kind of just chucking it away into a bin. But with tumors, sometimes they're able to kind of evade the immune system. Perfect. So we might go into a bit of basic cancer biology. So if you can see what's happened in this cell, we kind of can see there's a really big difference between what we call a malignant cell and what we consider a normal cell. So we've seen that we have abnormal cell growth, which is consistent with the definition of cancer, and then it leads to your cancer cells being able to move around. So I guess two key definitions to know, two key words to know, is the word angiogenesis and the word metastasis, which I don't think is on this slide. So angiogenesis basically refers to a new blood supply, but we'll go into that a bit later. And metastasis is basically what this cell is doing right now. It's kind of invading other cells. So if we look at these pictures, we can just see that what cancer does is that it's a lot of cell growth happening at once, which leads to uncontrollable cell growth. And these are what forms tumors, basically. And then we kind of have two types of tumors and two types of cancer cells. So we have a benign tumor and a malignant tumor. So based on these pictures, which one do you think is, I suppose, the more harmful tumor?
Malignant.
Yeah, perfect. And that's because it's kind of broken out of the cell. So it's been able to metastasize. So it's moved out of the cell and it's able to move to other parts of the body. So a tumor starting in the breast could go out into the blood supply and it could go into your lungs. Or I think it might be the other way around, but yeah. So tumors can spread through malignancy. With benign tumors, they're kind of confined to where they first form. So they're not able to move out of the cell. So even though they're still really abnormal and we don't really want benign tumors, they're less life-threatening than malignant tumors. And then if you look at these pictures, what do you guys think if one of this will be malignant tumor and one of this is a benign tumor? So which one do you think this one is?
The benign one?
Perfect, yeah. So it doesn't look, it's still pretty abnormal, so it still is a tumor, but it's a little less, I suppose, as chaotic as the malignant tumor. Especially in the pictures, you can see this one has a lot of cell growth. And yeah, as we were saying before, mutations are basically what underlies what cancer cells are. Mutations are what leads to abnormal cell growth. And then, yeah, so the last topic we have is the hallmarks of cancer. So this is probably the most complicated thing I'll go through with you guys. So don't feel too worried if you don't understand it straight away. It's just basically what we kind of say the properties of cancer cells are. So the hallmarks of cancer were introduced in 2000 by cancer biologists Robert Weinberg and Douglas Hanahan, and they describe the complexity of cancer behavior, and they kind of give it 6 properties, which kind of helps because if you think about it, if you only need to know 6 properties of cancer, it's a bit easier to tackle. But before we go into these 6 hallmarks, we should probably just go back to thinking about the cell cycle, because the cell cycle is kind of what helps cancer cells to start replicating. So as we were saying, we have the PMAP, so the M phase, and we have interphase where there's just DNA growing. So we kind of have an extra protective factor in the cell cycle, and we call these checkpoints. These checkpoints check for DNA damage, they check for cell growth, and they check for nutrients. But the thing about cancer cells is that they're able to bypass these checkpoints, because they're able to bypass these checkpoints, this can cause more growth and just less stopping. So these checkpoints can't really function as normal. Cancer cells kind of run their own cycle. They do this by sustaining growth signals. They can avoid stop signals. So we have what we call an oncogene. So our normal cells have this word called proto-oncogenes, and proto-oncogenes are what regulate cell growth. They're what tells our normal cells, oh, you should grow now so we can divide you into 2. But what the cancer cell is able to do, they're kind of able to mutate this proto-oncogene into an oncogene. And what the oncogene does is it says the cell to keep on growing and growing and growing. So it grows more than it really should, and it doesn't stop growing. So because of that, the cancer cells are able to sustain growth signals, and they're also able to evade stop signals. So we can also, in normal cells, we can stop cells from growing. And yeah, now we'll go into the 6 hallmarks, which are a little bit hard in my opinion, but I think you guys will get them down packed. But yeah, so sustained growth signaling. So as we were saying before, growth factors are able to control cell division. So these can be hormones in the form of proteins, and they can be released by cells, which signal other cells nearby to divide. And this signal is kind of vital. So as we were talking about signal transduction before, the reception part is what's the normal aspect of how our cells work. So signaling is considered normal. So cells use a code from the DNA to make the receptor be placed on the cell wall. And then when they bind, there's kind of a cascade of signals, so a transduction of events that allows the cell to enter the cell cycle and replicate. So this is the normal 3-step process we were talking about before. But the thing about cancer cells is that DNA damage kind of causes faults in the making of these growth receptors, and it allows them to be activated without the binding of the growth factor. So there's no reception to it. So they can really divide without being told to. So they're kind of bypassing that first step, and they're able to go to the second and third step, which we don't really want, because we always want to say when we want our cells to divide. We don't want them to start kind of dividing when we don't want them to. So because they're dividing, because they're replicating, the faulty DNA is passed onto the cells, and this in turn divides despite having no signal telling them to do so. And so the next hallmark is that it's kind of able to escape growth suppression. So tumor cells can ignore signals that stop them from dividing when they shouldn't. So damage to the production of tumor suppressor genes are able to allow the cells to continue through the checkpoints unchecked. The tumor suppressor genes is our body's own natural defense line to tumors. It's kind of saying, oh, let's suppress the tumor. But because the cancer cells are damaging these tumor suppressor genes, it stops them from working, and it leads to them once again being able to just divide uncontrollably. And then, yeah, the keyword we were talking about before is the word metastasis. So invasion and metastasis. So as we know, cells of different tissues are kind of supported by their surround, so by connective tissues. And that surround kind of forms an extracellular matrix. So this is kind of the stuff outside the cell, and it keeps the cell having structure, it keeps the cell having stability. But with metastasis is the fact that cancer cells can spread to other tissues and organs. And this is really important in determining whether cancer is curable and treatable. So if we have a lot of metastasis, if the cancer's moved to all parts of your body, it's probably more life-threatening and a little less treatable than a cancer benign tumor that stayed in one part of the body. So when activating invasion and metastasis and inducing angiogenesis, this kind of creates a really good environment for the cancer cell to keep on dividing. So remember, angiogenesis is just that creating of a new blood supply. So our blood is basically where we get red blood cells. It gives us oxygen. So think if the cancer cells have their own blood supply, they're getting their own nutrients and they're able to just survive on their own, basically. Yeah, so another hallmark is replicative immortality. So on our chromosomes, we have this kind of special aspect called telomeres. So with the telomeres, you can think of it as a shoelace. So we have a shoelace, and you know that tip of the shoelace that kind of stops, I suppose, it from fraying? That's basically what the telomeres does. It kind of holds the chromosome in place. But in our normal cells, the telomere shortens over time. So obviously with the shoelace, sometimes it starts fraying because it gets old. So that's the same with telomeres. Over time, our telomeres shorten, as you can see in this one. But with the cancer cell, what the cancer cell is able to do is that it's able to kind of implement this protein called telomerase, and it's basically able to tell the chromosome to just start making more of these like genes on the telomere. So it kind of lengthens the telomere as well. So what we can say about these cancer cells now is that the cancer cells are immortal. They're able to survive for a really long time because in normal cells, once the telomeres shorten, your cells are just told basically that it's time to undergo apoptosis. But if the telomeres are always lengthened, then your cells don't realize that they're old, that they shouldn't be there. So we don't undergo apoptosis. So your cancer cell is considered to be immortal. And then we have, angiogenesis, its own blood supply. So it's for what the word angiogenesis means. It's the formation of new vessels, which kind of supplies the cancer cell with its own nutrients, and that allows the cancer cell to grow and divide. So it's like how we eat, I suppose like a 5-year-old eats a lot of food to get taller. It's the same thing with angiogenesis. We're giving a lot of blood supply to the cancer cell to get bigger and bigger. And then as we were saying before, avoiding apoptosis. So what the cancer cells, they're immortal, they're able to avoid apoptosis, and they do this by kind of avoiding the P53. So P53 is an enzyme that kind of just tells the P53 is P53 is a protein and it signals apoptosis. What cancer cells do is that they mutate the P53 protein and it stops it from working. So the P53 can't really signal for apoptosis in cancer cells. And then another hallmark, so we're getting there, there's only like 2 more left. Yeah, there is 6, so there's quite a bit of them. But what the cancer cells can do is that they can avoid immune destruction. So basically our normal immune cells, when we get the flu, we kind of try and destroy the pathogen, right? We try and destroy it because we know it's abnormal. So our cancer cells, they're not really self cells, so they're still more abnormal to our normal cells. So theoretically, if you think about it, cancer cells, our immune system should be able to destroy them. But the thing about cancer cells is that they've evolved in that they can kind of really avoid the cells that destroy them. So they avoid T cells, they avoid B cells, they avoid all these immune cells. And they do this by kind of making themselves look a little bit like normal cells. So some medications can cause suppression of the immune system. So treating people with overactive diseases can cause immunity, and these people are more at risk of cancer because they don't have enough immune cells to destroy the cancer cells. But yeah, there's still a little bit more research going into how avoiding immune destruction kind of works. And then the final one is genomic instability, tumor promoting, and inflammation. So genomic instability, as we know, is that cancer cells continuously accumulate mutations and pass these mutations on to daughter cells. And they do this at a much faster rate than our normal cells because we're not always constantly dividing in the same rate as cancer cells. So the genome of cancer cells is kind of associated with chromosomal instability as they lose telomere protection through replication. So this instability can be in amplification and deletion of chromosomal segments, and this further contributes to a loss of genomic integrity, allowing tumors to form. And then in terms of inflammation, tumors are also known to create a pro-inflammatory environment by surrounding themselves with immune cells. So what's kind of interesting about this is that you might think that immune cells can destroy the tumor, but research has shown that this kind of pro-inflammatory environment is kind of associated with increasing production of molecules and factors that promote cancer cell growth. So even though we have a lot of immune cells around them, because what inflammation does is they bring a lot of immune cells to one area, even though we have the immune cells, they're not able to carry out their normal function. And that's what makes cancer so developed. It's able to surpass a lot of our normal kind of barriers to abnormal things happening in the cell. So yeah, in summary, not all tumors are cancerous. So we have benign, which aren't as life-threatening, and we have malignant. Just to note about benign is that over time, there is a possibility that they can become malignant. But yeah, with that, not all damages to the DNA will lead cells to become cancerous because we have the repair system and we have the process of apoptosis that can occur. And then cancer cells have specific properties that occur during their development that allow them to really persist in our body, even though we should be able to fight them because of all these properties. They're really more developed. And yeah, I'll pass it on to Hasna. Yeah, if you have any questions, just chuck them in the chat. And then Hasna and I also have a Google Forms quiz that you guys can do in your spare time as well. I'll stop sharing and then Hasna can share.
Yep. Thanks, Hashini. So I'll just share. Okay, so hopefully you guys can see that. Okay, so my name's Hasna and I'll be taking you through the HPV specific topics, so like the screening and the prevention, and also treatment of cancer, especially cervical cancer. Okay. All right, so what is HPV? HPV stands for the human papillomavirus. So it is a virus just like how COVID is a virus. Okay, that means it's a non-living kind of thing that needs to exist in our own cells. Okay, so what they do is they hijack our own machinery, okay, like they hijack our ribosomes to make proteins. So you can kind of see an image on the right, and generally patients will come in for HPV, and what they'll have is like warts. Okay, that's the normal presentation of HPV. But there's actually more than 100 different genotypes of HPV. So this just means that there are more than 100 different types, like strains of HPV. Okay, just like how COVID has the normal form of COVID and Delta. All right, so on that bottom dot point there, we can see that there are different groups of HPV infections, so they can each cause different things. So some of the common ones are the cutaneous warts. These are caused by HPV 1 and 2, genital warts as well, and genital cancer. So HPV 16 and 18 generally cause genital cancer. So we have what's called low risk and high risk HPV. So if you have low risk HPV, you're generally asymptomatic, meaning you're not going to have any symptoms. You're not even going to notice it's there. So you can kind of think of this as like a mouse has infected your house or infested your house. Sometimes you won't even notice. Okay? Sometimes you might have some warts from the low risk HPV. We still classify this as low risk because it's not as serious as a cancer. Okay, so moving on to high risk HPV. This is when you're infected with HPV 16 or 18 or one of the other high-risk HPV types. Okay, this often causes cervical cancer. So you can kind of imagine it like your house has been infested with mice, and every time the mice replicate, they get bigger and nastier every time. Just a small content warning for the next slide. I'm going to be showing you an image of like HPV symptoms. So on the left-hand side, you can see cutaneous warts. So cutaneous just means like on the skin, and this is generally caused by low-risk HPV, so HPV 1 and 2. Right, on the right-hand side, you can see a cancerous lesion of the cervix. So this is what high-risk HPV looks like. Okay, So let's talk about the epidemiology of HPV. Okay, so even though you might associate HPV and cervical cancer with women or people who have cervixes, it's important to understand that HPV will infect both males and females. Okay? So even though only a person with the cervix can go on to develop cervical cancer, both males and females will be infected. Okay? So HPV spreads from just with skin-to-skin contact, but it's primarily associated with sexual intercourse. Most people, up to 90% of people, will be infected at some point in their lives, but we can usually clear it naturally, especially if we have a really good functioning immune system. All right, so this means that people who are unable to clear the infection, like if they are immunocompromised, then they're more at risk for developing cancer. Okay, so how exactly does HPV infect us? All right, so if you come into contact with someone who has a HPV wart, oftentimes the HPV particles will actually be on the wart itself, right? So if you get some HPV virus particles on your skin, you're not actually infected yet, right? So if it just brushes onto your skin, the HPV particles aren't able to actually go inside your body and cause damage. This is because our skin is generally intact, meaning it's not damaged in a way that viruses can come in. Okay. So this means that if our skin is damaged, like if it's cut or burnt, the virus can actually enter our skin and infect us. Okay, so it's important to know, especially because HPV is a sexually transmitted disease, that during intercourse, we can have microabrasions, which are just when the topmost layer of our skin, so the topmost layer is called the epidermis, so that is removed, so it's like abrased off. And then we have the dermis, which is the layer underneath, that is exposed. So people might not even realize that they have microabrasions on their skin, but the virus can still enter and infect them. Okay. Also, the mucous membranes, like the mouth and the lips, are more easily infected by the virus as well. Okay, so let's talk about how HPV causes the disease. All right, so you can see on the right-hand side a kind of schematic of the cell. All right, so if you have HPV particles, remember they have to be inside your cells to actually live. So if they're inside the cells, what they do is they produce proteins, viral proteins, using our own cell machinery, okay, our ribosomes. And so they produce what's called E6 and E7. The E is just because they're produced quite early in the cell life cycle, on the virus life cycle. And what these proteins do is they disrupt our own cell proliferation. Okay, so they make a cell more likely to actually divide and proliferate. So this means you're going to essentially have more and more cells being developed because of the E6 and E7 proteins. Right? This is why we have warts, right? It's a cell growth. Okay, it's also why we have the cancer, which is abnormal cell growth. Okay, so just a question to think about. What protein type is most likely being affected by E6 and E7? Is it A, oncogenes, B, tumor suppressors, C, antibodies, or D, transport proteins? Does anyone want to guess? Okay, we're going to see in the next slide. So it's actually tumor suppressor genes. Right, so what we can see here are the E6 and E7 proteins. Right, what they do is they meet up with our tumor suppressor proteins like P53 and RB. Okay, and what they do is they degrade our own proteins. Okay, so usually these proteins stop our cell from dividing. Okay, so you can kind of imagine our P53 and RB like locks. Okay, so what they do is they lock the door of cell division, meaning the cell cannot divide if we notice that something's gone wrong in the process of cell division. And E6 and E7, what they do is they melt the key down. So we can't lock that door anymore. It's permanently open and our cells cannot stop dividing. All right, so this slide is basically showing you the different types of HPV, the different types of cancers caused by HPV. So the rest of this talk is focusing on cervical cancer, but it's really important to note that HPV causes not just cervical cancer, it causes anal, vulvar, vaginal, penile, and oral cancer as well. So this has certain implications on who should actually vaccinate against HPV. Okay. So let's talk about cervical cancer, and to do that, we have to orient ourselves to the anatomy of like a proper cervix or healthy cervix, right? Okay, so what you can see is past the vagina. The cervix lies between the uterus and the vagina. It's like the opening of the uterus, okay? So it's like the C-section here. Okay, so cervical cancer is the abnormal growth of cells in the lining of the cervix. So even though cervical cancer is the growth of abnormal cells, not all abnormal cells are cancerous. Okay, but the more abnormalities there are, the more likely it is to become cancer. So most cases of cervical cancer is actually caused by the high-risk HPV types, the HPV 16 and 18. Okay, so that's something to think about. Yet, what can we call abnormal cells that aren't cancerous yet? And we'll see on the next slide. Oh, not this slide, another one. Okay, but in terms of epidemiology, I really want you guys to think like not just about these specific statistics, but do these statistics on this slide really correlate to all demographics? Like, does this apply to rural people, rural populations or urban populations? Is HPV more common in certain races or cultures? Okay. So other risk factors apart from the high-risk HPV, which is the most important risk factor. Smoking is another big risk factor. So smoking is a risk factor for a lot of other cancers as well. Other important risk factors include more than 5 years on the oral contraceptive pill. If you're immunocompromised as well, you're unable to clear that HPV infection. So it is chronic and persistent. Okay. Also, if you've been exposed to something called DES, which is an artificial form of estrogen, it used to be prescribed to pregnant women to prevent miscarriage up until 1970, but it's not prescribed anymore. Okay, so that the children with cervixes who were born to that mother were like seen to get cervical cancer at much higher rates. That's why they banned it. So I'm aware that the screening test, itself might not be too relevant for your research or your topic. So I'm just gonna fly through this part. So what we do is we look for HPV DNA in the cervix. Okay, it's important to know that the screening only tests for 14 different types, so not all of the 100 genotypes that we talked about before. Okay, so who does the screening? So anyone with the cervix age 25 to 74, and they come in once every 5 years. So it's just something to think about as well. What are the barriers towards being screened? And you might have noticed on the previous slide, that last dot point there was we also screen the people who are vaccinated. Okay, it's really important to screen the people who are vaccinated because our vaccine doesn't cover every single type of HPV genotype. Okay, remember genotypes are the different mutations of HPV. Okay, so even if you're still vaccinated, you still have a likelihood of contracting a HPV type that wasn't included in the vaccination and going on to develop cervical cancer. Okay, so just quickly, how does the screening test actually work? We insert a cervical brush and we kind of extract some of the epithelium tissue that lines the cervix. So the procedure might be a little bit uncomfortable, but it should not be painful. So once we get those tissue cells, we can test them for HPV DNA. If it comes back negative, that's great. All right, see you in 5 years. If it comes back positive, we keep on searching. Okay, we keep on trying to confirm that diagnosis. So we refer them for a colposcopy, which is just a procedure to examine the cervix for signs of cancer. And we can also refer them for biopsy, which is a surgery that we're going to talk about in the next slide. Okay, so this is what the results of a biopsy will look like. Essentially, what we do is we get cells from here, the lining of the cervix, we can look at them. And so the cells here are called stratified squamous epithelium. The stratified just means there's many layers, and the squamous refers to the shape. Okay, that's why cervical cancer is called squamous carcinoma. Okay, so what we can see, we'll compare normal epithelium to something like cervical cancer. Okay, so what we can see is the dots in the middle are the nucleuses. Okay, and the big difference between here and this last one is that our cervical cancer nucleuses are very much enlarged. They're very much darker, and what we can also see is the cell shape is vastly different as well. Right, another big thing to notice here is that the cells can actually leave their kind of house, their basement membrane, and they can go into the bloodstream and spread around the body. Okay, so that's called metastasis, which is what Hashini talked to you about before. All right, so just to quickly explain L-cell and H-cell. L-cell is called low-grade squamous intraepithelial lesion, and it's a very common result on a pap smear or a cervical screening test. And H-cell can become cancer, but it's not yet cancer. So just quickly, the history, we implemented the HPV vaccination program in 2007, but it was only in 2013 that we actually included teenage boys. And our most recent vaccine, the Gardasil 9, was introduced in 2018. It actually prevents against 9 different HPV types, so not even close to the 100 different genotypes present. Okay, but we are like targeting the most high-risk types. Okay, so how exactly does the HPV vaccine work? All right, so what we can see here is this is kind of like schematic of the HPV virus. Okay, so the vaccine introduces our body to a modified form of the virus. Okay, it's kind of like if HPV was an army and the full army came to attack us. Right, so imagine the army is carrying a flag, and the flag announced who the army was. Right, what the vaccine does is it doesn't give us the army part; it just gives us the flag. Okay, so what we can do is we can identify the flag, we can tell who it is, so our immune system does that, and then it can tell how to best fight off this infection. Okay. So that later, when we actually see the army itself, we can go, oh, I recognize that flag, I know how to beat it, and then we can clear the infection more quickly. Okay. So understanding how a vaccine works is a common point of misinformation, so it's important to think about that as well. All right, so what we can see here on the left image is an antibody. So antibodies are produced by B cells, okay? And once we have immunological memory, so once you've been vaccinated, we produce antibodies much more quicker than we would have if we had not been vaccinated. Okay, and what we can see here is the antibodies are able to fight against the virus. Okay, so just quickly, the challenges of the HPV vaccine. We have over 100 different types, so you might be infected with a type that we haven't even vaccinated against. Okay, but success of the HPV vaccine, it's prevented up to 90% of cervical cancers. All right, so very common questions. Why exactly would we target high school students? Well, cervical cancer, or sorry, HPV is a sexually transmitted disease. And high school students are, at the age that they're more likely to start engaging in that behavior, and that's why we vaccinate at that age. Okay, and immunizing boys is also really good, so we can get herd immunity. Okay, and they're also susceptible to like penile and oral and anal cancer as well, so good to immunize them as well. Okay, so now we can move on to treatments. Okay, so if I told you, let's talk about the treatments of cancer, what people would generally think of is how we're going to cure the cancer. All right, but that's not always the case. So sometimes the treatments of cancer, sometimes we can't actually cure the cancer. Sometimes it is metastatic, it's gone to other parts of the body. And unfortunately then, some of the treatments for cancer would just involve prolonging survival or minimizing, alleviating the symptoms. So I'm going to talk about the different aims of treatment. Okay. So hopefully you remember what Hashini talked to you about. Okay, so this is an example of a local cancer, right? It's only one specific spot. But if we have a metastatic malignancy, so that means it's gone throughout the whole body, okay, well, we can see it's more widespread. So obviously this one is going to be more challenging to actually treat. Okay. So we'll talk about local therapies first. There's two types I'm going to be talking to you about, local therapies and systemic therapies. Okay, so local therapy is like if we had cervical cancer, okay, and a local therapy is surgery or radiotherapy. So we'll look at that now. So there's a couple of different aims of surgery. All right, so the first one is diagnostic surgery. This is when we can take out a cancerous lesion or just a part of the body that's been affected by cancer, or we can test it. So what we do is we take it out. Right? So what they've done is they've marked a border to cut it out, and they examine it under a microscope. And what we can see is the increased nucleus size, the darker staining nucleuses, to actually diagnose if a person has cancer. Okay. The next type is curative surgery. So this is the type that people will generally think about. Right? So what we have here in the dark pink is abnormal tissue. So we can imagine that this is cervical cancer. Right? So what they do in curative surgery is they cut around it, right? But what we want to do is cut a little bit more than what we can see as cancers. This just helps us make sure that we put all of the cancer out. Okay, so preventative surgery. You might have heard of the Angelina Jolie effect. So a couple of years ago, Angelina Jolie discovered that she had the BRCA mutation for breast cancer, and she decided she didn't want to risk developing breast cancer later in life. And so what she did was actually remove both of her breasts to ensure that she would never develop breast cancer. Okay. And so it's a form of prevention, just removing the organ that would be associated with the cancer. Okay, so if you were to remove your uterus, for example, that is also preventative surgery. Okay, so this is reconstructive surgery. It's just when you do surgery to make the body part look like it used to before you did one of the other types of surgery. And this is palliative surgery. This is the quality of life surgery. If a patient does have malignancies around their body, we unfortunately cannot cut out every single instance of cancer that they have. Okay, so what we do is we perform some surgery. We might perform like get some of the cancer out just so they experience fewer symptoms and a better quality of life. Okay, so this is the second local therapy. So it's radiotherapy. Okay, we have different types. So curative is exactly what it sounds like. Neoadjuvant therapy is when we perform radiotherapy before the surgery. Okay, so the aim of this is to shrink the tumor so that we can actually remove it with surgery better. All right, adjuvant therapy is after surgery. It's when we want to make sure that we kill all the metastatic cells around the body. Okay, and palliative surgery is similar to what palliative radiotherapy is similar to what palliative surgery was. It's shrinking a tumor so that we relieve symptoms for the patient. Okay, so this is just what the radiotherapy looks like. So we can see external radiotherapy. What a patient does is lie down here, and then the radiotherapy machine will twist and rotate around them. Okay. We also have internal radiotherapy. It's more targeted towards a specific tissue. Okay. Systemic therapy. So this is the last subject that we'll go through today. Okay, so this is when we actually target cells all around the body. Okay, so we usually use this for metastatic or malignant cancers, which have already gone throughout the body. We'll also use this for chemotherapy. For blood cancers. Okay, so 4 types that we'll talk about. Okay, the first type is chemotherapy. So if you remember back to Hashini's talk on the cell cycle, okay, so G1 is just when the cell is kind of replicating some, S phase is when the cell is replicating its DNA, and M phase here with the other cross is when the cell is actually dividing. Right, so chemotherapy usually works on either the S phase or the M phase. So it either stops the DNA from replicating or it stops cells from dividing. Okay, importantly, chemotherapy will not just affect the cancer cells, it's going to affect every single cell in the body. Okay. And this is the reason why a lot of chemo patients will lose their hair, right? Because hair is fastly replicating and it's going to be affected by chemotherapy as well. Okay. So hormonal therapy, we use this for cancers of the sex organs, like the breast or the prostate cancer. So in this case, they use our hormones like estrogen to actually grow. So what we do is we stop estrogen from getting to the site of the cancer, so the cancer stops growing as fast. Okay. So in surgery, radiotherapy, chemotherapy, and hormonal therapy, we are still able. We will still be targeting our normal cells. We're still going to have that collateral damage, especially with chemotherapy, okay, and hormonal therapy as well. Okay, so the idea that we want to target cancer cells instead of our normal cells is the basis of targeted molecular therapy. So what we do is we target the specific genetic mutations of cancer. Okay, and just this last one, immunotherapy. It just boosts our natural immune system. So Hashini talked to you previously about how cancer can evade our immune system. What immunotherapy does is it makes our immune system so much stronger, and it helps our own cells fight off cancer. And you might have heard of checkpoint inhibitors. That's a common form of immunotherapy. Okay. So after a patient has gone through treatment, they can come in for checkups and we can describe how they're going. So if they have a stable disease, that's when the cancer is staying the same size, or it might be growing a little bit. Okay. Progression is when the cancer is not responding to treatment and it becomes worse or it becomes metastatic. Right, metastatic is when it's spreading throughout the body. Okay, partial remission is when some but not all of the cancer signs disappear. Complete remission is when all of the detectable cancer disappears. Okay, but it's important to know that even in complete remission, the cancer might still be in the body. Okay, and even like a couple of years later, the patient might come in and be complaining of some cancer symptoms, and this is when recurrence has occurred. Right, recurrence is just when the cancer has come back after a period of time that we couldn't actually detect any cancer cells. Okay. So that's all we actually have for you today. I'll stop sharing. So Hashini and I had some Google Forms quizzes that I'm going to send you the link. So they're just if you want to consolidate your knowledge. So we'll put them into the chat.
Awesome. Thank you so much, Hasna and Hashini, for your knowledge and explaining it all so clearly. If anybody has any questions that you'd like to ask, please feel free to unmute and ask, or you can send your question into the chat.
If they have any other questions, I'd just put my email in the chat and they can definitely email me if they want.
Awesome. If that's all, then I'll pass over to Amy to conclude for tonight.
Yep. So thanks, Hasna and Hashini and all of our participants today for coming along. We very much appreciate everyone's patience with today's Zoom difficulties. But despite this, I hope everyone has gotten something insightful out of Hasna and Hashini's fantastic workshop presentation. And just a gentle reminder to our participants that registrations close this Friday, the 27th of August, and submissions are due Sunday, the 5th of September. Thanks, everyone, and we'll see you soon.
Thank you.
Thanks, guys.
Thank you.